Okay, so update your journal. Here's what we got. Ignore this right here. Okay. Pause it, update it. What we did on Monday was we did this Amoeba Sister video worksheet. I've got a copy of it for you, and we're gonna actually go over it in this video, so you can go over it with us. You can either do it beforehand or just do it with us. Cell transport notes, they're up on the board. I'm about to go over them. We were supposed to take a quiz, but I'm pushing it back till tomorrow. We're still gonna put it in there. And then we're doing a card sort lab, which is on the desks. Okay. So here are the notes. Okay. I did use this right here to summarize like how transport occurs, but you can check out last week's video for me to do that. And then here are the notes that we put in our notebook on 210. Okay, guys. Okay, the cell membrane. Now, this yellow part over here, the cell membrane is made up of lipids, but they also have protein tunnels, okay? It does not make sense for DNA to be in there. It should be in the nucleus, or glucose or carbs. So the cell membrane maintains homeostasis. It's made up of lipids and proteins. Proteins make up the channels. And it's said to be selectively permeable, which means only certain things pass through. Passive transport, remember, molecules cross membrane without using energy, so it's anything that would not involve energy, which is small particles. That's just regular diffusion, when particles spread out from high to low. Water, which is the movement of, or water diffusion, which is osmosis. And when anything is shown going from high concentration to low concentration, it's going with the flow. It's going into the less concentrated area. So pretty much anything that doesn't use energy. Now, sometimes you go with the flow, but you need the help of protein channels like these. As long as you're going with the flow, then you are just doing diffusion, but it's called facilitated. Facilitate means to help. You can't, like for example, glucose. Glucose can't pass through the membrane, so they need help. They'll go through one of these channels, but as long as they're going from a high to a low, so they're going to the low space, then they're using passive transport. If they would be pumped into a greater, like a crowded space, then it'd be active. Okay, good examples of that. Does anybody have their cards on their desk? Okay. Good images of passive would be something like this, where you're going from a high number to a low number. Okay, this is simple diffusion. It simply goes across the membrane. If there was a protein channel right here, it would just be facilitated diffusion. It's still diffusing from high to low. It would just need to be facilitated through a protein channel. Okay. Next notes are active transport. Okay. So active transport, this is moving molecules across the membrane using energy, specifically ATP energy. If you ever see ATP on there, they're probably talking about active transport. Now, when larger molecules take energy, or larger molecules also take energy. Endocytosis, endo meaning in, this is when something moves in. Usually the cell membrane has to open up and engulf it. It takes a lot of energy to move the cell membrane like this. Then exocytosis, this is when you expel something, and also the cell membrane takes a lot of energy doing this. Pretty much any time when you go from low and fight your way into high, going against the crowd or against the gradient, this is active. So here's some examples. Okay, this is an example of the cell membrane opening up, filling, basically engulfing and bringing it in. This takes a lot of energy. Okay, so that was endocytosis. Here's an example of exocytosis. Moving something to the surface and expelling it. Takes a lot of energy, okay? Anytime you see, this actually says ATP energy, and look at, it's fighting its way into the crowd, okay? Fighting its way. Okay, guys, so let's go over your worksheet. Okay, so question number 1, you had to sketch the lipid bilayer and label the polar heads and the non-polar tails. Okay, so this is what I wanted to see. I want you to draw at least 3 lipids. So these guys are fatty acid glycerol molecules. These are what one lipid looks like. And then I want you to draw at least 3. Okay, you're making a layer of lipids. Now, the cell membrane is called a bilayer. Bi meaning 2, bicycle 2 wheels, tricycle 3 wheels. So we're making a bilayer. Okay, so it just backs up to each other. Okay, this looks like a head. Okay, so here are the polar heads. Okay, and then here are the non-polar tails. Make sure those are both labeled for number 1. Okay, number 2, what was the answer? We did this one together. What is simple diffusion? When molecules move from high to low. So, simple diffusion. Here's the cell membrane. Okay, when molecules move from a crowded to a less crowded. Okay, high concentration to low concentration. They don't have to fight their way across the membrane. They don't have to fight their way into this space. Awesome. Okay, question number 3. How many did y'all circle? 3. Anybody else get any? How many did they circle?
I may have circled
2. Okay.
I didn't circle any. I just crossed out the wrong ones.
So how many did you leave as correct?
3.
3? Okay, let's check it out. So guys, they asked us only circle what's true about simple diffusion. So simple diffusion is just no energy crossing the membrane. They didn't say facilitated diffusion, so they're not talking about protein channels, and they're not talking about energy, which is active. A, it's a form of passive transport. Yeah. Yes, I agree. Passive transport, no energy. Simple diffusion, no energy. Make sure A is circled. B, molecules travel with the concentration gradient. Yes, so the key word is with the gradient and not against it. So yes, circle B. C, this is how glucose travels across the membrane. Video said glucose needs a protein tunnel at least. It can be active depending on which direction it goes. D, this is how oxygen and carbon dioxide travel across. Yes, and it's because they're small. Okay, they're only 2 molecules. So water is CO2, that's 3. Oxygen, O2, that's 2. Carbon dioxide, CO2, that's 3. Little bitty. They could just go right through. They'll just pass right through. E. This transport is typical of large molecules. No, that's active transport. Okay, number 4. Okay, this would be good for number 4. Okay, so you guys have the selectively permeable, semi-permeable membrane drawn, and there's an arrow that goes this way. First thing it asked you to label was where the high and the low should be. So if the arrow is going this way, and they're asking you to show passive transport, you need to be going from high to low, and you need to be going like with the flow and not against, okay? You had to draw 10. I would accept if you drew like 7 on one side and 3 on the other. You could do 8 and 2, 9 and 1. You could even do 6 and 4, but can you do 5 and 5?
No.
No, and you definitely cannot have more on the low side than the high side. Good. Okay, we did 5 together. Endocytosis and exocytosis are both active. Give a scenario where a cell may need to perform endo. So why would we need to bring something in?
They need to engulf a large starch or something for food.
Like
Good. So bring in something large, and I like how Jack said, for example, like a starch. Good. So bringing in something good that the cell needs. Number 7, give a scenario where a cell may need to perform exo. Why would we want to get rid of something super large?
Waste.
Waste. Good. Good. Okay. Backside, we did not do these bottom ones, so you don't have to either. Okay, I would say these last back ones are probably the hardest ones, and if you could get through these, then you can definitely do the quiz no problem and the test no problem. So, your answer choices are either simple diffusion, facilitated, which remember facilitated means with a protein channel, and active. So basically, simple no energy with a protein channel, energy. Okay, number 8. For water to travel across the cell membrane at a substantial rate. What does substantial mean?
Like at a noticeable, like
Noticeable,
lot.
good. So I would say there is a substantial amount of y'all that did not do your homework. Like a noticeable. Okay, so we're not just talking about a little water, we're talking about a substantial, a lot of water. The water molecules travel through protein channels known as aquaporins. So the fact that they mention protein channels, what is this? F facilitated, I agree. Change it if you get it wrong. Okay, number 9. While water molecules are polar, they are also very small. One fact not mentioned in the video is that water molecules are able to squeeze directly through phospholipid bilayer due to their small size. Small, no energy, what's that? S simple. 10. Charged ions are traveling through the cell membrane with the gradient. They don't really tell you what charged molecules, but they say with the gradient. So what'd y'all pick?
Yes.
S, I agree. Okay, 11. Cells lining the gut need to take in glucose, but at a certain time, the concentration of extracellular glucose is lower than intracellular, or than the concentration already stored in the cells. What does extracellular mean?
Outside.
Good. So here would be an example. And guys, you may have to draw examples just to see what we're doing. Okay, so they said here's a cell. We need to bring in glucose, but sometimes extracellular is lower than intracellular. What is it called when you have to push, pump molecules in against the gradient? Active, because it uses energy. Okay, then somebody read me 12.
At a certain time, glucose is in a high concentration outside of the cell and needs to travel through the membrane into the cell.
Good. So in number 12, they say glucose is in high concentration outside, needs to travel inside. Okay, so you're going with the flow, but glucose does need a protein tunnel, so this is not just simple, it's facilitated. I agree. Okay, yeah, I feel like they should have told you that on the last one. Okay, so tape that in your journal. Fold it in half, tape it in. I've got tape up here if it's a little bit jacked up. I got tape in my wheelie thing, and then we're gonna move on. So that's what we did yesterday, amoeba sister transport, the notes that were just on the board. We're gonna push back our quiz till tomorrow, which is Wednesday. And we're about to watch Amoeba Sisters Osmosis, just so we understand osmosis a little bit more for our quiz. And we're about to do a card sort. Should we take? Yeah. Okay, sorry I look like this. Remember, it's cozy day at school.
Something's on your tape.
I know, that tape is jacked up. Okay, so we're not going to watch this full video. We're also not going to do the worksheet. We're just going to watch bits and pieces that are going to help us. Osmosis can get really tricky. They can get really in-depth, like an AP concept. We don't need to know all that stuff. We're going to learn what we need to learn for the STAR test. So, we're going to take bits and pieces. Okay, so let's start.
You know what word gets thrown around a lot, but is actually pretty challenging to understand? Osmosis. But it does answer some great questions like, why would it be deadly to give someone an IV of pure water? Or why would a saltwater fish placed in fresh water experience major problems? We'll answer these questions as well as explain the process of osmosis. Now, first understand that osmosis is the movement of water, and that really is a type of diffusion. Often when you're talking about osmosis, you're talking about the movement of water through a semi-permeable membrane like a cell membrane. A cell membrane has openings small enough for water molecules to pass, but it will not allow larger things like salt molecules to freely pass, at least not without assistance. Osmosis is a type of passive transport, meaning that it doesn't require energy. I mentioned osmosis is a type of diffusion, and similar to what you might have learned about with diffusion, molecules, in this case water, travel from areas of high concentration of water to low concentration of water. But there's another way to think about water movement in osmosis. A low water concentration likely means there are a lot of solutes. Solutes are substances like salt or sugar that can be dissolved within a solvent, like water. Water has the tendency to move to areas where there's a high solute concentration, which means there's less water concentration. So if you want to easily figure out
So what we're gonna learn about is water goes to the party. We have to figure out where the solute particles are, not the water particles, but where's like the solutes? Where's the sugar? Where's the glucose? Where's the thing that they're talking about? And water's gonna go to the party, okay? So obviously the party's over there, water's going to the party.
where the water will travel, look to the side where there's a greater solute concentration. So let's imagine you have a box with a semi-permeable membrane in the center. And remember, this membrane will let tiny things.
So what does it mean to be semi-permeable?
Allows some things.
Selective, it allows some things but not everything. So remember, little things, oxygen, water, CO2, gases, some charged ions can just diffuse with the gradient. Bigger things need either protein channels or they need to be pumped with energy. Good.
Things pass through it like water and gases, but not large solutes like salt. We'll label one side of the box A and one side of the box B. If you have pure water on both sides of the box, those water molecules, they're moving. Water molecules, they like to move. But the net movement of water on the two sides is zero.
Okay, what that means is water is always moving in and out, but however much moves out, the same amount moves in. So even when we're talking about an isotonic cell, remember iso meant same. It was when it was perfect, the same amount went in, that came out. If we're talking in terms of like milliliters, if you have a cell, and guys, milliliters is so much bigger than what a cell would use. But if 2 milliliters went into a cell, how many would come out? 2. So the net was 0. So intake was +2, outtake is -2. So +2, -2, 0. So what they're saying is the net is always 0. But if I would say 0 water comes in or out, that would make you think it was not permeable. Nothing would come in and out, but it is. It's just whatever comes in, the same goes out. And this is only when it's in homeostasis, and this is when it's equilibrium, okay? Now we're gonna talk about when it's not in equilibrium.
Meaning the overall change in direction of movement is 0. Now let's imagine on side B, you dump a huge amount of salt there. So what?
Okay, how many salts? So the salts are the white squares. Those are the real particles. Those are the solute particles. How many particles are on this side?
4.
A.
9.
0. I don't see any salt particles.
Oh.
Okay, what about salt particles on B?
3.
3. So if water goes to the party, is it gonna move to side A or side B?
B.
B, because the party's over on B. More of the solutes are on B, okay? And that's because your main goal for osmosis is to dilute all of the high concentrated area, okay? So it just wants to go and dilute. So technically, if this was a box, this was side B water level, this was side A. If side A water went to side B, what would happen to the level in A? It would go down. and then B would go up, okay?
Which direction will the water move towards? A or B? The answer is B. This area has a high solute concentration. And remember, water moves to areas of higher solute concentration. You can almost think of the water as trying to equalize the concentrations, diluting side B. Once equilibrium is reached, the net movement of water on the two sides will be zero.
Think of it as,
The percent of water to solutes, you wanna have on one side, if there's none, it's 0, to the other side where it's practically 0.
yes, and I like how you said practically, because can you ever get an exact equal if there's no salts on this side or 3 on that side? No. Picture those salts as acid. Do you want some part of your body to be highly acidic? No. Dilute it as much as possible, and that's what your body does. So when water goes to the party, it goes and tries to dilute the particles to make it the same, okay? Now, if that's where we're gonna stop. Okay, now let's give some examples, some real world examples. Okay, it goes into this, but I'd rather draw it out here. Okay, so everybody look at the board, and I say water goes to the party. So if you're ever confused as to where the water travels, you put a star where the higher particle concentration is, and you move water there. So ready? I bought a fish from the pet store. I was so excited. I didn't even read about the fish. I didn't read the directions, and it was a saltwater fish. Well, I go home, put it in my fresh water straight from my sink. What's gonna happen? Okay, according to the laws of osmosis, first thing we do is we put a star where the more particles are. Where would I put the star? Inside the fish, outside the fish? Good. Where does water go? To the star. So water is gonna go to where the particles are to try to dilute all of this particles. What's gonna happen to this fish?
Low.
It will blow, yep, and blow up. Who remembers what it's called to swell up?
Hypo.
Hypotonic, good. So this is hypotonic. Okay, I don't wanna screw up, so then I go buy another fish. I think it's the same fish because they kind of look alike, but it's not, of course. I don't read the directions. I get so excited, I bring it home, put this new fish, which is actually a freshwater fish, doesn't have any salt particles inside of it, but I put it in salt water. Now, where do I put my star? Where do I put my star for the particles?
The water.
In the water instead, and where does the water go? Leaves the fish,
Shrinks up, dies.
and yes, it shrivels up. Who remembers what it's called to shrivel?
Pertonic.
Good. Awesome. So that's real world applications why you can't do that. Now, if for some reason it was the exact, so if it was like 4 particles, let's say 6 particles on the outside, 6 particles on the inside, and if it was the same, whatever water would go out would come in, and this one would be isotonic. So the key is, especially on your quiz tomorrow, to count the number of particles and always determine where's the party at, put a star, water goes to that star. Okay, now does everybody have a lab in front of them? Okay, get out your lab. I'm gonna give you 5 minutes to work on this, and then we're going to go over it as a class. I'm going over it in the other room, that way I could finish this video. But basically, you have pictures and word scenarios. Just put whichever belong to the passive on the passive side, whichever belong to the active on the active side, and then we'll go over them. Okay, so let's go over this while they're taking the time to actually think it out. I'm going to give you all the answers. Okay. I'm just sorting the word ones with the picture ones. Okay. So passive, no energy, active energy. Water enters the nephron. Who cares about the nephron part? Water is passive. So water diffuses. Oxygen enters a muscle cell. Who cares about the muscle cell? Oxygen, it's small, passive. Carbon dioxide exits an alveoli. I don't even know what that is, but carbon dioxide is a small gas, so it's passive. Okay. A neuron releases neurotransmitters. So when a cell releases, I would say this is the same as expels. So this is exocytosis, which is active transport. White blood cell engulfs a bacterium, engulfing. This is endocytosis. Salt moves into a microvilli. See, they really need to give us some more information for these rest two. Glucose enters a cardiac cell. Salt into a microvilli. So I'm going to put salt on the active transport side because a lot of times we have to pump it in. But a lot of times glucose enters with facilitated diffusion, which means it goes with the flow, but it uses a protein channel. So we're going to put that under passive transport. Okay. Pictures. See, there's a picture of the cell and they say a bunch of O2s, oxygen, that's passive. Then we got this picture. And then above that bean shape, it has O2, CO2, H2O. All of those are passive. Okay. Now, this actually has ATP on there. Anything that uses ATP is active, uses energy. Okay. This one also, it's going against the gradient. It's going from 1 into 5 using ATP energy. So that's active. This is endocytosis. You see everything's coming in, active transport. Exo, they're expelling it, kind of looks like a pimple. Exo, active transport. Okay, even though this one doesn't say active, I would never give you something that looked this tricky, but this does look like it's a pump, so we're gonna put it on the active side. And then this one, these look like just particles diffusing across the membrane with the flow. So that's passive, okay? What you're gonna do is you can pause the video here, and on the page that says card lab, we are going to write 1, 2, 3, these 4 bullets on the passive side. We're gonna write these 3 on the active side, and then we're gonna pick one picture to draw with it, okay? Same with over here, one picture to draw with it. So on this page, you can put the heading, passive transport, 1, 2, 3, 4 bullets and a picture. Active, 1, 2, 3 bullets, and then pick a picture.